In this video, we will discuss that the mitochondrial DNA in evolution. Let's see how. Let's see. Okay. So if you talk about the evolution, yes. So first point we are having that the evolution, which means the evolving things, the evolve. Okay, the mitochondrial DNA like evolve. So mitochondrial DNA is basically having the unique pattern of inheritance. Okay, unique pattern of inheritance. What is inheritance? As you guys all know, inheritance means that there is the overall contact, which means the total transferring of the genes into the in offsprings, like mother to child. Okay, so this is also known as the uniparental inheritance. Okay, this is the general topic we are talking about, unipattern inheritance. What is unipattern? Which means that the mitochondrial DNA is basically transferred from mother to child. That is called the uniparental inheritance. So there was the no recombination, which means that because of the one parent, because of the haploid cells were produced because of the one parent. If we talk about, there was the transfer from the mother to child. If we talk about we are having the changes is occurring between the, we are having the types, such as you can take the example of the nuclear DNA of human beings. Okay, so they are biparental. They are biparental. They are biparental. Let's see how. In which it means that two parents is involved, but here we were having the uniparental, means single parent. Okay, so this is the example of the human body. For example, A B C, A B C, A B C, and A B and C. Here you can see that we are having the exchangement of the gene, where you can say that as a recombination point. Recombination point. Here the crossing over is occurring, which means exchangement of gene between the homologous chromosome between the parent 1, this is the parent 1 mother, and parent 2, between the parent 1 and parent 2. Okay, so there is the exchangement of the gene. Here you can see that we are having that B is basically downward. Okay, B is getting changed and C is also getting changed. B and C crossing over occur, this thing occur. Okay, so to maintain that, we are having the endonuclease, which basically cut that gene, and we are having the ligase, which basically attach the segments, and this segment basically produce. Okay, these both endonuclease and ligase we are calling it the recombinase, because we are calling that. Okay, so here you can see that here the changing is occurring, the crossing over is done, the changing is occurring, but here you can see that these both are not changed, these both are same, which means that the recombination is not occurring here. Here the recombination is not occurring because of the linkage. What is linkage? Linkage is basically you can say the crossing over which is not breaking. Okay, not breaking of the crossing over. Okay, so the genes will be same in the offspring because of the linkage. They were non-linkage, they were break easily, and they basically change in the segment, they basically change. So this is what, this is the biparental inheritance. Biparental inheritance. So this is the example of the human body, which is biparental. Okay, but if you talk about the general, so here we are having the haploid cell. In haploid cell there will be the overall contact of in the offspring, which will be the uniparental inheritance. Just one parent will be there. Okay, so here we have seen that the mitochondrial DNA is basically helping is estimated to help in evolution. How to see the species are close or not, that how species are closely related to each other. So mitochondrial DNA is basically having a estimate or having the help in the evolution to see the species are closed or not. Okay, so they basically tell about the haploid and you can say that the biparental or uniparental. So this was the thing was in the uniparental, and this was the biparental in the human beings. So this was about the mitochondrial DNA in the evolution. This was the general topic we have discussed here. So in the next video we will discuss about the bacterial habitat.